Good morning, everyone. I would like to thank the organizer for giving me this opportunity to talk about the recent work. So this work, I studied the role of activity and dissipation in the precise beating via a simple model. And this work mainly done by student Subhajit Gupta and our collaborator is Dephased Chaudhary from IFP. And this is, we put it in the archive. So what cilia and flagella exist on the surface of cells. So for example, here there are few examples. The cilia are micron-sized filaments that beat continuously, and here you have paramecium, opalina, volvox. So they don't just beat; they beat synchronously. So here you see the synchronous beating of cilia, and this kind of beating helps them efficient movement in the fluid or to create efficient flow. So cilia doesn't show the coordinate beating, but it also the beatings are very precise. So today I'm going to focus on this part, the precise beating of cilia. So this is the complex structure of the cilia. You know that the filament consists of many microtubules. So doublets, like 9+2 microtubule doublets, and then they have also motor proteins and collective behavior of this motor protein can bend this cilia, and then you have see this beating. So this is the complex beating pattern. So and this is the kind of typical length scale, velocity scale, and so what is important here, these beatings are very precise. So what does it mean by this? So these are the experimental results. If you think of the deterministic oscillation, something beating means some quantity is oscillating in time. And if you think of deterministic oscillation, so then if you plot its autocorrelation function, then it never decays. So the quality is infinite. Okay, so because of the noise, it can be because of the thermal fluctuation or active fluctuation, you have some damping in autocorrelation function. So you have two scale, one is oscillation scale and one is damping scale, and the quality factor is the ratio of oscillation, this damping scale versus divided by the oscillation period. So and this number 38 or this quality factor lies like 38 to 200, and you know that this is huge for those oscillation. So in this talk, I focus on that what is the role of activity and the dissipation. So because to beat actively, so they need activity because it is in the viscous medium, and also you have dissipation due to the viscous medium. So this is the simple model we studied. So this is called Rohr model. So this model is proposed 2003, and then it is also experimentally realized 2010. And this model quietly used to study the collective behavior of the cilia. So here we use this model to study the precision of cilia beating. So here what you have, two harmonic potential, red and blue, and one bead, microbead is so doing a Brownian motion under this one of the potential, and then then once it reaches a particular location, for example plus A or minus A, so we switch off one potential, this red potential, and switch on the blue potential. So then it goes here, and then again it moves in the downhill, and then once it reaches here, we switch on the other potential. So in this way, we create the sustained oscillation in this case. So you have two length scale in this problem, or the distance between two potential, that is mu, and then you have the oscillation is restricted between A and minus A and A, we call it amplitude. And you have, so this thing, the stiffness of the potential is K. So switching is effectively is kind of like you are pumping some energy. So pumping energy, the difference between these two potential is K mu A. So basically this, so this is the activity energy. So that means if you increase a mu, K or A, so it will, you are increasing the activity of the cilia, and you have the dissipation at d gamma. And so overdamped motion of these things is given by this, and with delta correlated noise, in the case of thermal noise, you have d gamma equals to kb, theta is the temperature. So we want to, so this is the typical oscillation you see in this case of Rohr model. So displacement is doing like this, noisy oscillation, and if you plot the autocorrelation function, it decays. So this is, so you can fit the autocorrelation function is this decaying periodic function, and you have time scale t, which tells you the time period, and this is the coherence time. And if you take the ratio, this is called quality factor. And for this oscillation, the quality factor turns out to be 3. So then we check how this quality factor varies with different activity and dissipative parameters. So if you change the amplitude, what you see that at a particular optimal value of A, you have maxima. So this is a kind of non-trivial, because you are changing the activity and increasing the activity, but still you are getting, it is not monotonically increasing, it is decreasing. But if you change for a given if you change different mu, so you see larger the mu, the better the oscillation. And here you have larger the K, that means the activity, better the oscillation. So, and here we have plot this noise strength. So larger the noise, poorer the oscillation. So quality is decreasing. And here we plot it with the viscosity drag coefficient. So if you have, and if you are using thermal noise, the quality doesn't depend on the viscosity. So the one thing is very important here, that all the cases we see some non-monotonic behavior, and if we scale this thing with this scaled, so we have a length scale. You remember that mu, so which is the distance between two potential. If you scale it, x-axis and y-axis, I scale it. This is d gamma is the energy for the dissipation, and k mu square is typical energy for activity. So you see nice collapse, and this collapse exists only when you have this noise is small, and it follows like this kind of thing. So we tried to understand the reason behind this analytically. So for that, we consider this precision as we want to understand measuring as like FPT noise. So what is the first passage problem here? So basically, you are starting from minus A, and only one potential, and it is going down, and we are looking for the time to reach plus A. So this is the first passage time, and this is noisy because it is doing Brownian motion over here, and for that, you know that you can, so basically we are trying to say that this first passage noise is creating the noisy oscillation. So, so the relation, we are trying to understand the relation between this noise. So actually you can quantify this thing as quality factor, alternative definition of quality factor, that is fluctuation in current, where current is this by the time it takes, which is the first passage time, and at the small noise, it turns out to be 1 by CV square. So Q and first passage noise, so this is the relative fluctuation in time are related by inversely proportional, and this is true only for small noise. And so then we use this, you know that this is, now the problem become very simple, the Brownian motion in a harmonic potential, and this is exactly known. But the thing is that the formula is really complicated with error function and double integrals, so we don't have analytical insight from them. So what we did, we know that oscillations are very precise, so we can do some kind of small noise approximation. So if we do the small noise approximation, then we can find the noise in time is related to noise in position. And finding noise in position is comparatively very, very easy in this algebra problem. So this is the, you can find, and using this formula, you can get a nice formula for the relative noise in the first passage time. And so, so this explains this scaling for quality factor, and also this also goes as non-monotonically. So this is the thing. Yes, okay. So this is the numerical analytical match, and it is again violating for larger noise. Okay, so I'm done. So this is the what we see for real cilia, new system, new data, that they see that with activity change, some kind of data is showing. This is some wild type and mutant. Maybe there is a non-monotonicity. Still, it is not clear. So maybe a new experiment can say that whether you can see non-monotonicity with activity or not. Okay, so yeah, thank you for your attention. Yeah.
So I just wanted to ask that, okay, you have a model, yes. But why this, meaning the fact that how are you relating to the actual system with the kind of activity that you are proposing?
See, the actual system is really, so you have filaments and real complex beating. So I don't know actual system.
Meaning
I know that
this particular fact that it reaches X and then something switches on. So is there some biological?
Yeah, okay, so the reason is that 2 sigma, because this is forward and so and reverse stroke. So that's why it is like 2 potential I took. So you have forward stroke and reverse stroke, and it is like simplest, but because it took care of 2 strokes, so then that's why I was taking. Yeah.
Hi. So in general, the cilia, the beat pattern of cilia depends on the surrounding fluid.
Yes.
But in your model, it looks like you're not taking care of any of that.
No, this is the viscous fluid, right? So the drag is...
For example, what I meant was, for example, chemotaxis.
Yeah.
So the beat pattern changes depending on the medium, the process of chemotaxis. Okay, the cilia sense the external chemical gradient, and the body tries to move towards the target. So that information is not included in you. No. Why?
So this is the simplest model where we can get analytical insight.
Cilia has sensors.
Yeah. So we wanted to get the analytical insight, which is rare. We don't have like how. So first the simplest model we start. Then we can do.
My question, a straightforward question is like how realistic your model is. That's my question. Straightforward. How realistic your model is.
Realistic model is people are
your
doing.
Model. How realistic it is.
So.
I see that. What I see is like there's a beat pattern, there's a change in the potential. That's absolutely fine. But is it the best? Realistic. Is it the model?
You can say it is not realistic, but one can do experiments. So and they did it, and so this experiment is realizable, and people already did. And we have theories. Many people are studying theoretical and computational and experimental study on this. So it is important to study this model. Yeah. So complex filament, we can do it later. Thank you.
So actually, is this overdamped or underdamped?
Overdamped.
So what I have seen that amplitude is decaying, but the periodicity is maintained. So that actually happens for underdamped case, right?
What? Amplitude is?
Decreasing, but the periodicity is maintained. That actually happens for underdamped case. So where you have shown the graph, with time. A few slides previous.
Okay.
Yeah.
Yes.
This one. Yeah. With time, AT versus T.
Yes.
The amplitude is decreasing, but the periodicity is
So
maintained. That
this is
actually happens for
this is
under.
autocorrelation function. This is the oscillation.
I mean, it has happened. You have shown it earlier also. I mean, there is similar.
So, yeah, so
similar
the autocorrelation function is decreasing, but this is the oscillation. That is like amplitude stays between this point to this point, plus A to minus A all the time. Maybe we can
Okay,
talk later.
but at the beginning you have shown a slide, right?
Yeah, so that is also autocorrelation
one?
function, some phase
Oh, okay,
correlation.
okay,
So
that's
that is
fine.
okay.
Okay, so let's thank our speaker.
Thank you.